Selecting an appropriate comparator, or positive control, in a human abuse potential study is essential for establishing the abuse potential of a test drug. The positive control should be an FDA-approved drug that is, if possible, pharmacologically similar to the test drug or elicits similar psychoactive effects, such as sedation or stimulation. It is also required that the positive control be scheduled under the Controlled Substances Act. This is crucial because the control drug must have established psychoactive effects that help define the benchmark for abuse potential. Selecting a positive control isn't always straightforward, particularly when the test drug has a novel mechanism of action. In these cases, it may be challenging to find an approved scheduled drug that perfectly mirrors the test drug's potential effects. For example, a completely new class of compounds may behave very differently than anything currently on the market. In such cases, sponsors should explore alternative positive control options, drugs that may not be an exact match in mechanism of action, but elicit similar psychoactive effects. The key here is to justify the selection of these alternatives based on their ability to produce comparable subjective effects to those of the test drug. When selecting the dose for the positive control, ideally the choice should be informed by previous human abuse potential studies, where those specific doses have demonstrated clear, positive subjective effects. If the positive control has not been used in prior HAP studies, it may be necessary to conduct an initial dose selection study. This allows researchers to identify the lowest dose that produces positive subjective effects that are strong enough to distinguish from placebo, ensuring that the dose selected for the HAP study will be capable of eliciting the desired pharmacological response. In summary, selecting the right positive control for a HAP study requires careful consideration of the test drug's mechanism of action, its observed psychoactive effects, if any, and the availability of suitable scheduled controls. In cases where a novel drug presents challenges in comparator selection, it's essential to justify the use of alternative drugs that can produce similar psychoactive effects. Lastly, dose selection should be informed by previous data, and when needed, an additional dose-finding study can provide the assurance that the correct dose is chosen for the HAP study.